artificial neural network works and in what way they actually recapitulate the way in which the brain functions. So first of all, just to be all on the same page, system neuroscience addresses the function of neural circuit in intact organisms. The goal of this research is to understand how the brain works, and in particular, how different ensembles or group of neurons that are scattered throughout the brain connect to each other, talk to each other, and by talking to each other, create functions like vision, like memory, like creating a motor response. So system neuroscience, if you want, is a sort of a very large, broad field of research that integrates different disciplines that necessarily go from experimental components to new way to analyze data, and you will see this on Friday, and computational technique to try to understand how the brain works with quite some engineering, because you will see, especially on Wednesday there is quite some sophisticated machines that we basically operate. So the question that we are trying, the overachieving question, if you want, that we are trying to answer is, how do different parts of the brain work together to produce the green ingredient of behavior? That is perception, cognition, and action. And for you to be on the same page as me with this, what I mean by these three terms is that perception is quite intuitive. You see something in the physical world, there is some sensory stimulus that impinges your sensory organs, your eye, your nose, your tongue, and you basically create a mental image of the thing that is outside of your brain, within your brain. So for example, in this case, you see a tennis table and you recognize that what you see is actually a tennis table. A tennis ball. The tennis ball might elicit in you the memory of something that has happened quite some time ago. That is Roger Federer winning a Wimbledon match in his most elegant way, which is an image that has happened sometimes in the past, but of which you have created a record because it has elicited in you some sort of response, for example, a pleasurable response, and that you keep for future recall. So this is what we create as cognition, starting from sensory inputs, creating mental representation of these sensory inputs, and filling this with meaning. Now, thinking back to that Wimbledon, because you like Federer and you, as me, are a strong supporter and you live in Basel, you might feel a little bit of happiness, right, at the memory of Federer playing in Wimbledon. And that might lead you to curl up your lips and to create a smile. That's what we call action. And in last instance, action is, in most cases, the way in which we interact with the world. So from perception to cognition to action, this is the job of system neuroscience. In last instance, what we want to do is to understand how information about experience is represented in the electrical and chemical signals the neuron use to talk to each other, what we refer to as the neural code that we're going to be talking about on Wednesday. We want to understand the computation that transforms this information as this information propagates along neural network and multiple brain areas. That's what we refer to with the name of neural dynamics. We want to understand how the brain uses this information to support behavior. So how information is encoded into neural circuits, but also how the brain is able to decode that information to make sense of it. And in last instance, we want to create in silico model that can reproduce cognitive function, because this is the last test of actually us having understood how the brain works, which is not an easy task all in all, but it's something that we are trying to approach, and especially in the past years have been really made possible. So again, to familiarize a little bit with the terminology that we are going to use during this week, I just want to make an analogy to what we are doing right now and what is going on right now with your computer and with you in your homes. So I'm talking, I'm emitting sounds that our microphones, in my case, my earbuds, are transforming into an electrical signal and to a radio signal that is transmitted to an antenna. What basically happens is that my voice is encoded and translated into a different language, from a sound wave, that is what I'm emitting with my mouth, into an electricity, and then into a radio wave, so that it can be transmitted to the computer and so on through your computers. And the wireless router at that point picks up the signal and then translates into yet another signal that is an electrical signal to transmit it through the air, through the internet, and through a varied Ethernet connection to a router, from where the message is further processed, divided in packages, sent to your computer, and you're able to hear a sound wave because the message, again, from electricity is transformed into a physical sound wave. So at every step of the way, message, so signal that carries information, is converted. We will use the word encoded to basically symbolize this conversion through specific computational passages from one physical medium to another, and then transmitted from sender and receiver, which is basically the same things that our neurons and our brain do. So the brain, the ear, picks up the sound wave and transforms the sound wave into an electrical signal. The electrical signal then is basically propagated from neuron to neuron across the nervous system, and at the moment of contact between two neurons, converting to yet another medium, which is a chemical medium through neurotransmission, that is important to transmit information from neuron to neuron. This chemical signal then is converted again into an electrical signal and can propagate along the network over and over and over again. And while this message goes through the networks and the different steps of the network of our brain, it gets meaning. It gets meaning of the fact that you're listening to me right now, and you might like what I say or not like what I say. You might remember the happy days in which you used to come to the Biocentrum, so you might remember the cat. That will allow you, hopefully not in the too distant future, to type down on your keyboard and ask me a question about what I just said. So these are all the different steps that we do. The important bottom point here is that information must be transformed from one media to the other in order to propagate along neural networks in our brain and allow you from sensing to attach meaning to it and to elicit, to force you or induce you to elicit a motor response. So this week we'll introduce you to concepts and methodology that are at the heart of system neuroscience. I will try as much as you do to make you learn from experience, which is the job of a scientist. That's why we're going to have multiple virtual laboratories with the software that I've suggested you in my email. And we will try to articulate our journey into system neuroscience in 5 different questions. The first is, what language do neurons speak? The second is, how do they talk to each other? The third is, what do they talk about and how we can understand what they talk about? That is the 4th question. And the 5th is, can we build an artificial brain? These are going to be the topic of the 5 days of this week. Today, we're going to talk about basic principle electrophysiology. Tomorrow, we're going to look into neural networks, biological neural networks. The 3rd day, on Wednesday, we're going to talk about neural coding, and we're going to tap into studying an animal's behavior. On the 4th day, we're going to understand how to manipulate neural activity in order to make hypotheses about what the neurons function and what actually, how they influence behavior. And on the last day, again, we're going to talk about topics that are a little bit more on the side of neurobiology per se, but that are very pervasive and important, that are artificial intelligence, machine learning, and deep neural networks. Okay? So the aim of the course is, I don't expect you to be expert in system neuroscience in 1 week. You probably could not be. If you are perfect, come and work with us. You are needed for neuroscience. But I really want to get a sense or an intuition of concepts that are really not only interesting, that might be interesting for you to study during your master, but also that are basically impacting your life on a daily basis. Why do your sense trick you when you see an image, for example, and why the internet goes crazy about this? Why does Google ask you to label images to authenticate yourself into websites? So please take this as an opportunity to learn something, to have fun while doing that, hopefully, to ask questions, make comments, and engage, which is basically what I'm really suggesting you. So this is the schedule of the course that I've given, that you have seen before. What I want to point out here is that all the exercises that you will do will have to be recorded. You will have to be a lab notebook. This is the way in which in the lab we actually document our experiments. We try to do it in the most thorough and clear way possible. Please be clear, because we have to read them. In fact, you have to turn in your lab notebook by the end of this week, which is next Sunday. Don't be scared about the amount of exercises that are on the assignments. I don't expect you to do all of them, but I expect you to reason about them. I mean, it's better to have few exercises in which you really understand and make prediction about what you will hopefully see, than not finishing everything in a mindless way. The last thing before we start with actually the lecture is participate. Please don't let me speak for 5 days without any human interaction. One, because your participation is going to be factored in as your grade. Second, because it's important to ask questions if you are not entirely sure about having understood some concepts. Again, you can ask questions anonymously. This is the code that you have to use. There was a mistake. I made a mistake. I had inverted these two. So please don't use the code that is in your slide, but use this one. Turn on your camera so that we can see each other. And the lecture will not be recorded. I know that a lot of people ask for that, but I think that you will get the slides. Recording the lecture just to have notions for a blog course, I don't think that is actually necessary. You will get all of this written anyway, but it's important that you focus and you engage right now. Okay. So this is the end. We can start with the first lecture, but if there is any question or comment, please do it now so that we can go on and talk about the fundamental principle in electrophysiology. No? So I guess everything is clear. Perfect. All right. So I have to say that during this course, it was very nice for me to actually go back into understanding a little bit of where we come from as neuroscientists. But I thought about starting this course about the fundamental principle in electrophysiology by telling you about year 1816, which was a very particular year because it was defined the year without summer. And that was actually quite true in the sense that in 1816, Mount Tambora was erupting, and the amount of ashes that Mount Tambora emitted in the atmosphere was so strong that the whole Earth plummeted in temperature, and there was basically not an increase in temperature in the whole period that goes between May to September. So Mary Shelley at that time was in Geneva at the house of their friends, Villa Diodati, and they come up with the idea of writing ghost stories to try to scare each other off. And she came with the inspiration of writing about a monster that was assembled out of dead bodies by a Dr. Frankenstein and brought to life by lightning. And that's how the creature, how she calls her, not Frankenstein, that was the doctor, but the creature came to life. This was actually something that is very relevant for neuroscience because the inspiration for this story was a treatise that had been published a few decades before by Luigi Galvani about electricity and the way in which electricity is inducing the twitching of muscles in a dead frog. So Galvani was using his way of basically like his electrodes to induce a twitch in the frog-like muscle. And what he saw is that you could induce contraction if you applied electricity to the nerve fibers, but not if you applied electricity directly to the muscle fibers. So what he ingeniously deduced from that is that the brain produces electrical signals that are able to control the body through nerves. And this was actually sort of a paradigm shift for the way people thought electricity would work. They thought that there was this ether that would go through our body and that would come basically like directly onto the muscles. He discovered that electricity is indeed the language of the brain and is the way the brain talks and communicates within itself, not to the rest of the body. But who is speaking this language? Speaking this language that was a mystery. How is the brain really created? At that moment, people thought that the brain was basically a full organ, that there was no distinction in self. People had to wait still a few decades until 1904, actually a whole century in that case, to understand that the brain is actually created by a complex network of individual identities, cells that are called neurons. And this was Santiago Ramón y Cajal, who was a remarkable scientist. He had to stop his study to go and fight in Cuba. In Cuba, he got malaria, and basically he survived malaria, although he was almost dead, came back to Spain and revolutionized science forever. He really is the father of modern neuroscience. I'm telling you this because you will see that there is a lot of stories of resilience in which very harsh condition actually sparks creativity in neuroscience. And it's actually quite inspiring during this particular and difficult time. What Cajal was able to visualize were neurons. And he could already describe the neurons as very particular cells because they're very polarized with very distinct features. You find that neurons have a dendrites, the neurons have a soma, that they have an axon, a long protrusion that basically can reach very far away from the soma. And then there is an axonal arborization, which is full of small buttons, which we call presynaptic terminal. But one of the most ingenious things that he actually deduced without any proof of that, and that's the amazing part of his intellect, is that the messages that neurons relate to each other flows from the dendrites through the axon, through the somatic area. So that at the level of the axon and the presynaptic terminal, it can be transmitted to another neurons and so on in our neural networks. So now we know what is the language. Now we know who speaks this language, but what does information assume in the brain? What is these messages? What is the message composed of? To understand again what the message is composed of, we have to wait 30 years until Hodgkin and Huxley recorded the first action potential, which is an electrical impulse that propagates through axon and that is used to communicate between one neuron to the other. Hodgkin and Huxley also had an amazing life in the sense that they made this discovery in 1939. Shortly after Hitler invaded Poland, they had to leave their lab to fight in World War II. At the end of the World War II, they came back to the lab and they actually continued their studies. So you see, nothing could stop neuroscience. Anyway, action potential are electrical signal by which neurons receive, analyze, and convey information. These signals are all or nothing events. It means that if you start an action potential, the action potential will go through this basically like shape, which is change in membrane potential. And it's always going to be the same throughout the nervous system. So even though action potential can be initiated by a great variety of events in the environment, for example, light, mechanical contacts, and odorants, a pressure wave, that is the sound wave that we were talking before, action potential are highly stereotypes. They all look the same, and they convey information about whatever they convey information about based on the neurons that emits them. And this is a very important principle in neuroscience. Another thing that is true is that neuroscientists are badass. They cannot be stopped by malaria, they're not be stopped by World War II or confinement. So here, to go back to the action potential, we see a key principle in brain function. The information that is conveyed by the action potential is always the same, but it depends on the neurons and the pathway through which the action potential travels through. What I showed you before is that you have perception, cognition, and action. If you were able to record the message they transmitted between neurons at these different steps, you will always find the same type of shape of the action potential. But the identity of the information and the meaning of the information is actually conveyed by which neuron are talking to each other to basically like either perceive the tennis ball, recall the image of Federer, or induce the curl of your lips into a smile. So we need to know more about this message, and is going to be the topic of today. We're going to understand how neurons generate electrical signal, how neurons decide when to fire an action potential, and how we can listen to action potential in awake animals. All right? Okay, so let's continue with the first question. How does a neuron generate an electrical signal? Well, the neuron can generate an electrical signal because the cell maintains a difference in potential between the either side of the membrane. What it means is that the concentration of ions, positive and negatively charged ions, is actually not even at the two sides of the membrane, of the neuron's membrane, so that the extracellular side, there is a high concentration of positively charged ions, and at the internal side, there is a high concentration of negatively charged ions. So in the end, if we were able to stick an electrode into a neuron and record this difference in potential between the inside of the cell and the outside of the cell, we would find immediately that the potential would drop to negative values because within the cell, we have a higher concentration of negative ions and charges, right? And would drop more or less to a value of -65. So this is called the resting membrane potential, and it's quite important concept because you will see it's something that allows neurons to actually transmit information to each other. So different cells, of course, have different resting potential. They can range from -40 to -80. This depends a lot from the type of cell that you're looking at, but let's say that we keep -65 for now as a sort of a working value, okay? So you see here a principle that is very important. The membrane of a neuron is electrically charged, and even in resting conditions. But how does you create a membrane potential, right? How do you make sure that this difference in charge is kept and maintained in the brain? Well, the resting membrane potential results from two factors. One, there is an unequal distribution of electrically charged ions, especially sodium and potassium, which are positively charged, and one negatively charged ion, which is chloride, which are the three ions that we will talk mostly about today. The second thing is that the membrane is selectively permeable. So you can make it more permeable to sodium or to potassium or to chloride in a controlled fashion. So in the end, don't pay attention yet to these arrows, because we're going to talk about these dynamics in a little bit. What I want you to get is that if you look at the extracellular side of the neuron, intracellular side of the neuron, you will find a very high concentration of potassium within the cell, but not outside of the cell. You will find a very high concentration of sodium outside the cell and a very high concentration of chloride outside the cell as well. So you have a difference in concentration of these ions that is kept like that in order to create a membrane, resting membrane potential, and is modified in order to create an action potential. The signaling in the brain, in fact, depends on the ability of these neurons to respond to stimuli, and we will see then how, with changes in this electrical potential in flows of ion across the membrane. Okay? The membrane potential is generated by two different forces. On one hand, you have an unequal distribution of charged ion, which is created by specific proteins that actively transport ions from an area of high concentration to an area of low concentration. And you have channels, other proteins in the membrane, that instead are able, when open, to dissipate this gradient and to allow ions to move from areas of high concentration to areas of low concentration. So in the end, ion transporters move ions against the gradient, while ion channels allow ions to move following their gradient. Now, ion transporters are move a solute or an ion through the membrane against its electrochemical gradient. They go from the low concentration to the high concentration. Since normally the ions would tend to move in the opposite direction, they need to use energy. And that's why creating a membrane, like an electrochemical gradient on the two sides of the membrane, is actually quite expensive. So this process is called active ion transport, because the ion pumps or ion transporters needs to receive energy to make their movement. So either in the form of ATP from within the cell, or in form of energy coming from the outside world, like light, or by using the energy that is created when you move another ion from an area of high concentration to an area of low concentration. What I want you to get from this slide is that ion transporters, also called pumps, they're actually quite important, because their work is the one that creates the gradient that the neurons use to communicate with each other, but they're also quite expensive proteins, because we need energy to actually make them do their job. Ion channels, on the other side, they are able to recognize specific ions, and you see the mechanism why. They have binding site within the ion itself that is able to bind specific ions given to their conformation and so on, and to create, when open, selective permeability through this to this particular ion. When they are open, they allow specific ion to move across the membrane following the electrochemical gradient. We're talking about the electrochemical gradient in a moment. So in the end, what I want you to have clear in mind is that you have two different classes of protein, the transporters and the channels, that are like the yin and yang of the brain, right? The transporters create the gradient, and the ion channels actually dissipate this gradient. And here I'm going to risk it, because you're very young, but who remembers this movie? Who knows what I'm talking about? See a lot of lost faces, so I'm feeling really old, but this is like when Miyagi in the Karate Kid was saying, given the box, taking out the box, right? One creates the gradient, the other one dissipates the gradients. 80s kids will know what I'm talking about. Anyway, what I just want to say is that opening a channel is actually quite an expensive thing, because you dissipate all the energy that the transporter has actually used to create the chemical gradient. So you need to regulate it quite tightly. And that's why you find different ways to regulate the opening of the ion channels, which can happen through three different mechanisms. The ion channel can change its conformation in a specific region. You need here that the inside of the channel opens up or closes down. You can create a general structural change in the channel so that the whole channel is basically open or blocked, or you can remove or add a small part of the protein that blocks or opens the channel. This is not quite as important as this slide here. That is what opens an ion channel. I told you before, ion channels opening is very expensive. You waste ATP, you waste energy given by light, you waste energy given by another ion. So you need to be able to tightly regulate it. And how can you regulate the opening of an ion channel? Well, you can induce the opening of the ion channel by binding to a ligand, by phosphorylating the part of the protein, most likely the intracellular part of the protein, by changing the voltage of the membrane, meaning that usually, as you see, you have an external positive charge, an internal, negative charge. When you have a change in the membrane potential so that the outside is negative and the inside is positive, the ions open. You can also use physical forces to open an ion channel. For example, you can have cytoskeleton filaments that are connected to the channel and are able to open when they stretch. Or you can use other stimuli coming from outside, and in particular light, in order to induce a change in conformation that open the channel. As I said, if the channel would be open all the time, they would continuously dissipate the work that the ion pumps are doing. So we need a way also to close the channels. And the way in which you close the channel is basically to revert the stimulus that has induced the opening. So you turn off the light in this case, you change again the membrane potential so that the inside is negative, you release the pressure applied here, you dephosphorylate this channel, or you remove by normal situation, like by unbinding the ligand that activated the opening of the channel in the first place. Okay, so I really need you to remember these two concepts, that you can open an ion channel with a ligand, with a change in voltage of the membrane, with an external stimulus that can be either light or applying a certain sort of pressure. Okay, if you got this, we are good for the rest of this presentation at least. Cool. Any question at the moment? Otherwise, I'll go on and I'll tell you what happens when you open an ion channel. Perfect. When you open an ion channel, there are two forces that you are starting. So the membrane, first of all, becomes permeable to the ion that the ion channel is specific for. But there will be two forces that try to move the ions from within the cells to outside the cells and the other way around. On one hand, ions tend to move from a region of high concentration to a region of low concentration. This is the chemical gradient, right? So, for example, sodium very highly concentrated outside of the cell tend to go within the cell. At the same time, though, you remember ions are charged particles, and you have a difference in voltage between the inner and the outside of the cell. So because particles tend to move, or charges tend to move to the part of the dipole that is charged in the opposite way, so negative to positive and positive to negatives, there is a chemical force, an electrical force, that will act on these ions to move this through this direction, right? So in the end, what you have is that when you open an ion channel, there are two forces, that are diffusion and the electrical fields, that will drive the passage of ions through it. So the difference in concentration will drive the ions from the compartment with high concentration to low concentration. The difference in charge will drive the ions toward the compartment with the opposite charge. At the end, these two forces will start to balance themselves out. So the force and the chemical gradient and the drive of the chemical gradient and the drive of the electrical gradient will balance them out so that the ions reach a level of voltage and concentration that is called the equilibrium, that makes them not move anymore. So let's see what happens when you do this at the beginning, right? In a cell that is not electrically charged, you open a channel and ions will start to move from the region of high concentration to the low concentration, okay? If you add a charge to the membrane, like the one that you have in a neuron, positive outside, negative inside, this force that will move the positive ion charges will be even stronger, because it's not going to be only the chemical force, but also the electrical force, right? So they will move very much toward inside. Little by little, though, the change in the membrane potential, for which the inside is going to be more positive than the outside, is going to be trying to actually counteract this movement of positively charged ion to within the cell, because they start to repulse each other. So in the end, the chemical gradient and the electrical gradient will work in opposite direction and will balance each other out. Okay? This is what happens in a neuron. In a neuron, I told you before that there is a very high concentration of potassium within the cell and a high concentration of sodium outside of the cell and chloride outside of the cell, right? So when you open the channels, potassium is trying to go out. And that's because you basically, even if you have a positive charge outside, the different concentration will be high. The sodium will start to go in because, you know, the concentration and the electrical gradient will drive it in, and chloride will start to go in again, like potassium. There is a negative charge within here, but the chemical gradient is so high that they're basically just moving, right? So the chemical electrical forces will start to fight each other and to move these ions and to counteract each other. And remember what I said before, when the two forces are balanced out, the ions reach equilibrium and won't move anymore. And we can calculate when the neuron, actually the forces reach equilibrium through one of the few mathematical formula that you will see throughout the course, is the Nernst equation. I don't need you to understand the mathematical concept of this. I'm just going to tell you now just for your knowledge. But what I need you to understand is that the Nernst equation has a fixed number that depends from the temperature and the valence of the ions, basically how many positive or negative charge the ion has. But it's very important and very proportional to the difference in concentration of the ions between the outside and the inside of the cell, right? So this gives us the equilibrium potential for each ion, meaning the value of the membrane potential in which the total force on the ion is going to be 0. So this value is also called the reverse potential, because when the potential of the membrane crosses this value, the ion will start moving in the opposite direction of when it was moving before. So if it was going in, it will start to flow out. At that value, it's not going to move. Plugging in the numbers for a neuron, we find that the value of reversal potential for potassium is minus 85 millivolts. So it's basically like driving things even lower than the resting membrane potential, which is minus 65. For sodium is plus 58, and for chloride is actually minus 79. Why is this important? It's important because ions, when you open channels, will start to fight to bring the membrane potential to reach their own equilibrium, right? So sodium will start to go in because it wants to bring the membrane potential to plus 58. But if you would start for chloride, for example, it will try to go in order to make the inside of the neuron more negatively charged to reach minus 79. It's sort of a push and pull in which the ions work against each other.
One question.
Okay. Yeah.
What's normally the temperature?
Yeah, exactly. So our body is around 35, 36. So this is the normal temperature when we work. In the lab, there is a lot of different temperature conditioning which these things have to be considered. So again, usually we try to use physiological properties as much as possible, but this is another point that we actually need to consider, the temperature in which you do, because this value is depending on the temperature that is in the numerator here. There are channels that are sensitive to temperature, as you know, the one that actually make you feel the temperature. Okay, so this is cool, but how does this relate to the language of the brain that I told you before, to the action potential? The action potential is nothing less than the change of membrane potential over time. In this case, the change you see is around minus 60, closer to our working resting membrane potential of minus 65. At a certain point, you start to have a depolarization that brings the membrane potential toward very positive values. And then at start of decay toward more negative values, then you actually start an upper polarization phase that goes back to equilibrium when the action potential ends. So what we want to understand is that action potential and rapid depolarization and repolarization of the membrane that is driven by the motion of sodium and potassium across the membrane, and the interaction of any type of ion channel that you have that can be basically like activated by ligand, by stretch, by light, by any other mechanical force, and the voltage-gated ions that are the ion channels that are on the membrane. So two ions, sodium and potassium, two type of channel, ligand gated or stretch or light or whatever. But anyway, regulated by some external force and the voltage gated channels. Okay. The key ingredient for the action potential, in fact, are these three type of channel: the ligand gated sodium channel, the voltage gated sodium channels, and the voltage gated potassium channels. These three are important to generate the action potential, and we're going to see now how. Okay, are we clear with the electrochemical gradients, the forces trying to move ions one side of the other side of the membrane, and why this is important for the way in which neurons talk to each other? I assume yes. All right. Okay. So let's start at rest, right? We put our electrode within the axon, we can record the voltage of the membrane, and we see that the channels are closed. I mean, this is correct, let's say 80%. There are some channels that are open, the neuron is leaky. But the pump has created a gradient between the two sides of the membrane, right? There is a high concentration of sodium outside, a high concentration of potassium inside. There is an electrical gradient that says that the membrane potential is at -65. Okay? Okay, now we have the first step. There are some sodium channels. In our case, let's say that these are ligand gated, that respond to the binding of the ligand to the channel by opening. And this allows some sodium ions to flow inside the cell to try to depolarize the cell. He's fighting to get to +58, if you remember. This produces small depolarization of the membrane, right? That is basically like trying to bring the membrane to more positive values than before. At a certain point, the membrane potential reaches a threshold. And what is this threshold? The threshold is basically like the level of the membrane of a more positive membrane potential than the rest, in which the voltage-gated sodium channel take it as a hint to open up. So the membrane starts to rise because of the influx of sodium. It reached this threshold, at which point the voltage-gated sodium channel open, and that shoots immediately the membrane potential to very positive values that are peak value of 30, 50 milliwatts. These are the values in which the sodium will start to basically like have very little force to flow inside the cell. At this peak value, though, the sodium channels start to close because they inactivate, but the potassium channel starts to open, the voltage-gated potassium channel. That leads to a positive efflux of potassium from the inside of the cell to the outside of the cell. Because remember, potassium is highly concentrated inside, but also the reversal potassium of potassium is at -80 something. So basically what you want to do is to get out ions, potassium ions from within the cell so that the membrane goes back to a value that is very negative. The membrane voltage now drops very fast. The cells hyperpolarizes because not only do it go back to the values of -65 where it was before the beginning of the action potential, but it will also go a little bit lower because the potassium channel, which now is in command, tells, no, let's go to -85 because that's my reversal value, okay? So if we deconstruct what happened during an action potential, you have a first phase in which you have an increase of permeability to sodium, which produces a depolarization of the membrane, which is first slow, remember, the ligand-gated sodium channel, and then very fast, because the voltage-gated sodium channels kick in. These will try to push the potential of the membrane toward the sodium equilibrium potential, right? At the value of -40 or 50, though, this channel will inactivate and the potassium channel will open. And at that moment, a very strong force will be applied on potassium to get out of the cell, so that the membrane of the cell will go back to a negative value. This is the depolarization part of the membrane. They'll try to shoot back to the level of values of the potassium equilibrium potential through an hyperpolarization force. At this value, also the potassium channel will inactivate and the membrane potential will go back to baseline because the pump and the different ions that are open induce a coming back to the membrane. And this is the action potential. This is the curve that you saw before, recorded by Hodgkin and Axley in the axon of the screen. Okay, are we clear? I really want this to be engraved in your mind because this is going to be what you're going to be working on today in the lab. So you have a first phase, a stimulus, which induces a depolarization of the membrane. I here put the failed initiation because remember, in order for the voltage-gated sodium channel to open, the membrane potential need to cross a certain threshold, right? So if you don't induce the crossing of the threshold, nothing happens. The action potential actually doesn't happen. It's like a failed action potential. Okay, but if you cross the threshold, then you have a very fast depolarization phase. Sodium goes inside the cell. You reach a peak at which this channel inactivate, but the potassium channel open, which brings basically like a lot of potassium channel to go out of the cell, both because of their chemical and electrical gradient in that case, shoot down to a value of membrane potential, which is even lower than what it was before, to go back then to a resting state. Okay? So this is, in a nutshell, in a superficial way, what happens with an action potential. But what about chloride? I told you that there is a third ion in the equation, right? Chloride. Chloride is very highly concentrated outside of the cell. And in theory, it shouldn't go really within the cell because the cell is negatively charged. But because of the forces that I've told you before, if you plug the numbers of chloride into the Nernst equation, you will realize that the reversal potential of chloride, or the equilibrium potential of chloride, is actually lower than the resting membrane potential. So if you activate channel that makes the membrane more permeable to chloride, you don't get a depolarization, but you actually get an hyperpolarization. So the chloride is starting to go in, not too much, because, you know, I mean, this is not, there is still an electrical gradient between the inside and the outside of the cell, but still high enough that the membrane potential goes to more negative values, and this will have funny consequences that we will see in the next lecture. Okay, so these are the concepts that I wanted to express you right now. We are finishing like quite early, but I don't want to overdo the presentation, because it's hard to focus and keep focus for a long time. I just would like to have you like chance to discuss about this, ask me question about this, and about basically the gradients that we have on the two sides of the membrane, and how these gradients influence the way in which neurons communicate with each other. Question. Let's see, first of all, if someone has done a, there are no anonymous questions, so I guess we are gonna have all overt questions. So first of all, have you heard before about action potential and how neurons create action potential in courses, right? So you're known about that. And you've also known about the technique that you use to record this action potential, right? In a slide at least, which is, you know, patch clamping, voltage clamping, whatever you want to do. Well, this is really the basis of understanding how neurons talk to each other. We are now facing a new way that is, you know, having the action potential is just one part of the equation. You need to communicate to others what you are actually doing, and you need to be able to modulate this event of creating an action potential with a way that allows you basically to pick up signal from the outside world. So we're going to see in the next lecture, after break, how these action potential are initiated by the stimuli that happen in the world around us, and how they're used to communicate between two neurons. All right. Okay, so I think that we can also say that we come back like in by 10:30 if you don't have any question. I'm gonna be online. So Fiona told me that you guys had suggested that you really wanted to have chances to talk to the people, that is either me or the other people in the lab. I just want to say that, like, you know, if you feel the need to create a Google Calendar or Google Sheet to just book time to talk with us directly, do. But at the same time, this channel is always open, so you can log in and talk to each other, and you can log in and talk to me about, and at the same time, just send me a message. You can even create like a direct chat with me, so that it's not public. If you want to talk about something, ask some question, get some information about neuroscience or about anything else about your course of study. Okay? Great. So this was faster. I'll see you at 10:30. Good. Enjoy the break.